A client came to us 18 months into their Leuprolide program. PLGA sourced, Microspheres produced, and a burst release profile that looked more like a bolus injection than a depot formulation. No one had caught it because no one had asked the right questions at the polymer characterization stage. Hello, I'm Raghuvar Singh Parmar, Scientific Affairs Director at Resolve Mass Laboratories, where we design and execute end-to-end development programs for complex injectable generics from initial characterization through ANDA submission. Why Leuprolide Depot generics are so complex? Leuprolide Depot is not simply a difficult molecule in a complex formulation. It is three overlapping problems compounded into one regulatory challenge. You have a peptide that degrades under shear, temperature, and pH stress. You have a PLGA microsphere system where the polymer itself is the release mechanism. And you have an FDA bioequivalence framework that expects performance equivalence, not just compositional sameness. Meeting that standard requires integrating chemistry, process engineering, and regulatory strategy from day one. Most development programs underestimate at least one of those dimensions. The one that underestimates all three rarely reach submission intact. Reverse engineering of the reference product. Before you formulate a single batch, you decode the reference product. Reverse engineering is where your entire downstream strategy is shaped. You're trying to reconstruct a proprietary manufacturing history from a finished product, identifying a PLGA grade, estimating encapsulation efficiency, mapping surface morphology and particle size distribution without access to the innovator's process documentation. The analytical rigor you apply here directly limits how many costly developments iterations you'll need it later. Teams that shortcut this step routinely arrive at pilot scale with a formulation built on assumptions they haven't tested. PLGA characterization drives product performance. The polymer is where the science becomes exciting. Molecular weight sets release duration. The lactide to glycolide ratio governs the degradation kinetics. N group chemistry, acid terminated versus ester cap, shapes how your polymer interacts with the peptide and the formulation environment in ways that aren't always predictable from first principles. Batch to batch PLGA consistency is not a quality detail, it's a regulatory requirement. If your polymer grade drifts between development lots and commercial batches, your release profile drifts with it. That's not a deviation, that's a program level risk. Microsphere engineering matters. Microsphere engineering is where formulation science and manufacturing science have to operate as one integrated discipline. Particle size, encapsulation efficiency, internal porosity, none of these are independent variables. Solvent evaporation rate affects internal pore architecture. Pore architecture governs how drug migrates to the particle surface. Surface drug loading directly determines burst. Teams that treat microsphere development as a formulation activity and manufacturing as something to resolve later consistently find themselves rebuilding the program when they reach scale. One program we supported had a textbook particle size distribution at laboratory scale, tight, reproducible, exactly what a regulatory reviewer wants to see on the development report. At pilot scale, a subtle shift in the emulsification step widened that distribution just enough to alter surface area to volume ratio across the batch. Burst release doubled. It took a systematic analytical investigation to trace the problem back to a single process parameter that had never been identified as critical. That is depot development. The variable you didn't consider becomes the one that defines your timeline. Controlling burst release is critical. Burst release is the challenge that humbles even experienced teams. Surface associated drug releases immediately after injection. That is physics, not formulation failure. The real question is magnitude, because early drug release carries direct safety implications, and FDA expectations for Leuprolide are specific and non-negotiable. Emulsification's energy, temperature, solvent removal rate, each process condition directly shapes your burst profile. You cannot optimize this at late stage. Burst control has to be designed into the first prototype, because retrofitting it at the end of development costs time you don't have. Testing IVIVC and analytical tools. In vitro release methods for depot injectables are difficult to build and harder to defend. The method must be discriminating enough to detect meaningful formulation differences, and it must correlate with in vivo performance. That's the in vitro in vivo correlation expectation FDA holds for the product class. GPC for polymer molecular weight, SAM for particle morphology, DSC for thermal characterization of the polymer matrix. Each analytical method must be developed, qualified, validated, and ready to withstand regulatory scrutiny. At this stage, analytical development is not a support function, it's a core deliverable. Manufacturing and ANDA expectations. By the time you approach ANDA submission, every parameter that governs product performance must be locked, justified, and backed by development data. FDA will scrutinize controls around encapsulation efficiency, particle size, residual solvents, moisture content, and sterility assurance. Teams that have built integrated formulation and process understanding from the beginning arrive at submission with answers already written. Teams that separated those work streams, running them in parallel without genuine integration, discover late that their science and their CMC narrative don't quite align. So, three things define successful Leuprolide Depot development: a polymer system fully characterized before you process a single batch, formulation and manufacturing treated as one integrated problem, not two sequential work streams handed off at scale up, and analytical methods rigorous enough to detect what matters and compliant enough to satisfy what regulators require. Resolmass has been inside these challenges before, not as consultants at a distance, but as the team executing the science. So reach out to us for your program and we would be happy to discuss that. Thank you so much for watching this session. Goodbye for now. See you in the next one.